Where we can start. So it is my great pleasure to introduce to you Petr Sulc, also known as Petr Sulc. So Petr is well known in the field of DNA nanotechnology as one of co-authors of oxDNA program, the oxRNA, and the web servers that allows you to do visualization and set up simulations. So Petr got his bachelor degree in theoretical physics from a university in Prague, and then he got 2 master's degrees, both in esoteric fields, quantum science, quantum physics, and some numerical simulations. He did a brief stay at Los Alamos lab, after which he moved for his PhD studies at University of Oxford, where he contributed and co-wrote the oxDNA software. After that, he got a prestigious doctoral fellowship at the Rockefeller University, and in 2018, he became a faculty at Arizona State University. And today, we'll hear about coarse-grained modeling for nucleic acid nanotechnology.
Thank you, Alec, for the kind introduction and invitation. Very happy to be here and enjoyed all the meetings I had today. And I presume this can be quite informal, so feel free to stop me at any time. In front of this audience, it might be a bit redundant to introduce what DNA and RNA nanotechnology is, but just to make sure we are all on the same page. The experimental systems we are interested in modeling rely on designing DNA or RNA strands in such a way that they fold into a target shape, or they are devices that rely on interaction between multiple different DNA strands to perform a computation. Now, this field has been quite successful because A, DNA is relatively cheap as opposed to other materials you want to manipulate at nanoscale. It can be handled in molecular biology labs with typical equipment. The only expensive part there is to have visualization capabilities using atomic force microscopy, TEM, or cryo-EM. But the actual equipment to assemble these structures is relatively cheap. Buying the strands to assemble like one of the hallmark structures, which are the DNA origami, you are looking at about $1,000 to order the strands. What are these things good for is another question. So for a whole time, the field started in 1980s with Ned Seaman. It was all about proof of principle, just showing you can do more complex, more fancy structures in 2D and 3D, or that you can do computation and compute square root of the number of strands you input, etc. Now the field is gradually shifting towards applications, and I think there are 2 main directions where this is going. So one direction is basically medical direction. So people are using it for diagnostic purposes, or looking also for drug delivery and inhibition. The reason is that you can really position very precisely molecules, proteins, small molecules, aptamers onto the nanostructure. You basically control with 0.3 nanometer precision where the molecule will end up with respect to each other. The other direction, which is the one I'm also going to be talking about later today, because of the project we are working hard in my lab, is using it for about template assembly. So this is basically using it as a blueprint to position inorganic materials. And as opposed to chip manufacturing, which is all in 2D, this in principle would allow you to really, with very high precision, positions in 3D space. And I will mention some plausible applications of this later on. Those are just a few other applications that people have done in the past, like proof of principle. This is a DNA barrel, which only opens up if this aptamer binds to a specific protein expressed by in this case, it was leukemic cell. It opens up and exposes this protein, which is supposed to cause apoptosis of the cell. So this was like a big fancy paper in 2008, I think. It never went into human trials. It was a company started before the pandemic, which then bankrupted. So it's still not around the corner, I would say, but a lot of work is going into these directions. These are algorithmic self-assembly tiles, so you mix multiple DNA strands together, which self-assemble into these fractal shapes. And this is a blueprint of a Boolean circuit, and you can actually show you can implement the Boolean circuit just with strand displacement reaction, which I'm going to describe in more detail, just by interactions between DNA strands. So I introduced the DNA origami structure on my first slide. So this is a cartoon. This is not a simulation. This is illustrated the basic principle how these structures are designed. So we have a long scaffold strand, and we have these short staple strands of length between 15 to 30 typically nucleotides, and each of the staple strands binds 2 or 3 different distant regions on the scaffold strand. And depending on how you design which staple strands binds to what other region on the scaffold strand, you can assemble a desired 3D or 2D shape. Now, I'm a computer modeler by training, so I'm interested in getting computational insights into how these structures assemble and how they work. We understand the thermodynamics quite well. We know that A will pair with T and C will pair with G, and if you wait long enough, you should get the free energy minima of the system where all the base pairs are formed. It, of course, never works as easily, and we don't really understand the kinetics, how these structures assemble, and they do not always look like or function like you designed them. So I believe this is the only crowd I ever gave talk in front of that doesn't need convincing that simulations are useful. But I'm usually used to facing experimentalists, which always ask me what I can tell them that is useful for them. So I need to justify my existence for them. But the goal is, of course, to simulate these structures, and they present many difficulties. So you saw they are quite large. Like the origami side, origami has 6,300 or 6,378 base pairs. The assembly process, the typical protocol is several hours annealing. If you actually were a bit smarter about it and just annealed it at the right temperature, maybe you can get it down to 5 minutes, as Henrik Dietz has shown, but still, minutes is sort of infinity for our computational folks. And it also involves rare events. You see base pairs forming or breaking during these processes, and this is all quite difficult. There has been some very heroic effort done by people in this room and Alex's group and his postdocs students that did manage to get some very nice full atomistic simulations of some of these devices and structures. But our approach is a bit different. We decided to sacrifice the precision for speed, so we developed coarse-grained models. So coarse-graining in our case is done with what we would call a top-down approach. So rather than starting from full atomistic representation and grouping atoms together, we sort of just say, what do we want the model to reproduce, and what is the minimum representation that we can get to reproduce the behavior we want. So in our case, we represent the entire nucleotide of the 20 to 30 atoms by a single rigid body with multiple interaction sites. So this oxDNA model was originally developed by Tom Awdry during his PhD, and I joined when he was just finishing, and I took over some of the parameterization tasks and implementing it for CUDA and CPU simulation package. The general philosophy behind the model is the following. So the rigid body is basically a sphere, but the sphere has multiple different interaction sites. There's the backbone interaction site, which connects to the nucleotide on the same strand with basically Feeney spring potential. And there is the stacking and hydrogen bonding site, which interact with other sites through empirically designed potential. So our potentials are multiple of different functions. There's one function which depends on the distance. So you want the interaction site, for example, for hydrogen bonding, to be at the desired equilibrium distance, which corresponds to the width of the DNA helix. And then you also want the rigid bodies to be oriented to be coplanarily facing each other. There are additional terms which depend on the angle, and they are at their minimum of the energy potential if they are exactly coplanarily facing each other at the right distance. We also want the neighboring bases on the same strand to interact with stacking interaction, which wants them to stack, again, coplanarily, perfectly aligned on top of each other at certain equilibrium distance. Now, the equilibrium distance at which the two bases like to stack above each other is shorter than the distance which they want to have their backbones at. So I cannot stack and have my backbone like this because the stacking wants to be closer. But if I would bend like this, I would not satisfy the coplanar requirement. So the way to satisfy both stacking and backbone distances is to twist the molecule. So the fact that the molecule is twisted is an emergent property of the model as a competition of these two length scales. Now, this would cause the model to be left-handed or right-handed with equal probability. So we have additional fudge term, which switches off the interactions if you happen to twist in left-handed fashion. So this way, we force the strands to only stack in right-handed helix. There is also a cross-stack interaction, which stabilizes this interaction by its neighbor. And there is coaxial stack interaction, which is basically just stacking between bases which are not nearest neighbor on the same strand. So what I just described was oxDNA 1 version of the model. Yes.
I was always confused by this image, and I always thought that nucleotides are this, but you just say they are.
No, so this is just for the visualization purposes.
Actually, it's here.
So actually, the nucleotide is a sphere, which is centrally here. There's the backbone side and there's the hydrogen bonding side. The backbone side has around it excluded volume interaction. So the radius of this sphere roughly corresponds to excluded volume interaction of the backbone side. And then there is the hydrogen bonding side, which has also spherical excluded volume interaction around it. The reason why we draw it as ellipsoid is that it's easier for visual cue to see how they are oriented in space.
But in reality,
but in simulations, it's a sphere. And when you have two nucleotides in proximity, you calculate the distances between all the pairs of the interaction sites. You check that excluded volume doesn't overlap. And then you go through all these non-bonding potentials for their interactions.
So you don't have anything for AT versus CG.
So in the original version of the model, AT and CG were the same strength.
Yeah.
But what was half of my PhD was to actually reparameterizing the model to have the sequence-dependent interactions. And how is it the model parameterized? So the basic structure, the B helical structure, is basically set by hand just by setting the equilibrium positions of the interactions to be where we want them to be. The thermodynamic properties were obtained by running melting simulations. So you will basically run simulations which goes from double strand to single strand back and forth many times, and you get the free energy difference between bound and unbound state. From this, you can calculate melting temperature, which is the temperature at which you have 50% dissociated of all the strands. There are some small little caveats that actually you have to extrapolate to bulk from this box. Hide it under the carpet for now. But what is the 50% in a simulation box is not 50% in the bulk. You have to apply some thermodynamic corrections for it. And then we basically reparameterize the strength of these interactions so that the temperature at which you have 50% open closed corresponds to the one which was measured in experiments. We didn't go to all the experimental papers one by one, but we used something that is called Santa Lucia model, which is an effective fit to all the thermodynamic data. You basically enter the sequence and it gives you the predicted free energy difference between bound and unbound states. And it reproduces all the experiments within 2 to 3 Kelvin precision. So we treat this empirical model as an effective fit to experimental data. And then we had an automated simulated annealing algorithm which searches through all possible stacking strengths and hydrogen bonding strengths to reproduce the melting temperature of around 70,000 of different sequences that we used it to parameter. So that's how we go to thermodynamics. The next thing was also to get the mechanics reasonably well. So what we wanted, what is key process for the DNA technology is that single strand is very flexible, freely jointed chain pretty much, but DNA duplex behaves as an extensible worm-like chain. And this sort of came out to be emergent property of the model. You can modulate the percent length in the model by narrowing the potential. So how much you allow the angle to wiggle. So by basically setting the shape of the interaction, which modulates how much you want these two subsequent base pairs to be coplanarily aligned. So the model has 125 base pair persistent length. So if you don't know what present length is, if you just measure how do the normal, if you draw a normal vector to the midpoint of the base pairs, and you measure the correlations between subsequent vectors along the helix, and the correlation between subsequent vectors decays as exponential minus distance over present length. So DNA present length is like somewhat around 130 or so, or maybe up to 150. We are slightly below it, so the model is slightly more flexible than actual DNA duplex, but by not too much. In terms of interaction with ions, so the original version of the model didn't have any way to incorporate salt concentrations at all. The latest version, which we call oxDNA 2, implements the bihedral potential. And again, we took the duplex melting experiments which were done at different salt concentrations, and we parameterized the effective backbone charge as a function of the salt concentration to reproduce the melting temperature as seen in these experimental data. It is reasonably okay down to 0.2 molar. I wouldn't trust the model's representation of statistics below this number. So, as I mentioned, the key part which we wanted this model to reproduce was that two single strands are flexible, but when they meet each other, they hybridize and form a right-handed, reasonably rigid duplex. And it needs to be also reasonably fast. So 1 second of this video is about 1 second of a simulation on a single CPU. Those are some of the stuff which have been studied with the model. So this is when I started my PhD. My first project was to pull on single-stranded poly A and poly T and try to look at qualitative difference that the stacking can have on the force extension curves. It was a single CPU code. Now, with our colleagues from Italy, we were able to implement a CUDA version of the code. So our the most latest, not yet published data, we can simulate up to 2 million nucleotides on a single CUDA card for sampling simulations. So we had studied systems which would be basic biophysics property of DNA strands, but most of the applications have gone to nanostructure constructs. I also developed a model which is a younger cousin of the DNA model. This one is for RNA. So it follows the same coarse-graining philosophy. Again, single nucleotide is represented as a single rigid body. There are a few differences that I wanted to make. So one is that in most conditions, RNA duplex is of what you would call an A-form helix. So that means that if you draw the helical axis, it doesn't pass through the midpoints of the base pairs as it does in the B-form helix of DNA, but sort of the whole helix wraps around the helix, around the duplex wraps around the helix. And also the angle, if you draw a line through the base pairs, they have like inclination of about 15 degrees with respect to the axis of the RNA molecule. The model only supports Watson-Crick and wobble base pairing. It doesn't support any tertiary contacts, and it's again mostly designed for RNA nanotechnology applications. So RNA technology more or less follows the footsteps of DNA, except that it uses RNA as its basic building block. The example of published applications of the model include like studying RNA duplex under superhelical twisting stress. So it does reproduce the fact that if you start twisting RNA duplex, it forms these plectonemes. We studied few RNA nanostructures like RNA origami, RNA nanorings, or these RNA tiles which interact through kissing loop interactions. But the field of DNA technology is much bigger than the field of RNA nanotech, so our main focus still remains DNA nanotechnology. But since I did introduce the RNA model, the first application I'm going to talk about is with the RNA simulation. So this was the general introduction of the model. I'm happy to come back to it if you have further questions. But in the next section of the talk, I want to give few examples of what these models can be useful for and what we are using them or our collaborators are using them to study. So what really was our jackpot, I would say, is that we accidentally ran into Eric Winfrey, who's one of the big experimental / theoretical guys in the field of the nanotech at the conference, and he wanted us to simulate a strand displacement reaction. Now, that paper was already published back in 2013, and we did it with DNA, so it's a bit older work. So I'm going to talk about our recent work, which studies the strand displacement with RNA, but the mechanism of the reaction is the same. Basically, this is the key trick in all activity nanotechnology. What the displacement means is that you start from a gate strand, which is the green and red. The green is the incumbent and the red strand is the substrate. You see that the substrate has few extra base pairs, which is called the toehold. Now, at room temperature, for this particular number of base pairs, it's extremely unlikely that the green strand will spontaneously dissociate. However, if you introduce this invader strand, the blue one, which is fully complementary to the red, it will attach to the toehold, and then by random fluctuations, what will happen, you will sometimes break this base pair, and the blue one can jump in and replace it. And this process goes back and forth, back and forth. You can either dissociate and fall from the toehold if the toehold is too short, but if you wait long enough, the thermodynamically preferred state is the one where the blue strand kicks out the green one. And this one is much more stable than the start you started from, because you now have extra 4 base pairs, which the system didn't originally have. And you can show that you can then combine these gates to realize arbitrary logic operation, AND or XOR. I'm not going to go into the details, but basically the basic trick is that once this strand is released, it can take part in some other reactions, and taken together, they would act as OR and AND gates. In the actual experiment, the input is that this strand is either present or not. You would have multiple gates. And if you introduce the gate, the strand which opens the gate, it means 1. If you don't introduce it, it means 0. Then if this strand gets kicked out, at the very end gate of your cascade, you will have this strand which is fluorescently labeled. So if the strand is kicked out, it will start giving you a fluorescent signal. And whether the signal is or not present also means that the readout is 0 or 1. And you can have multiple different colors, so you can have multiple different readouts, and people are now quite clever at designing fairly complex circuits. I think that they actually managed to implement a neural network consisting of few neurons, which was like able to perform image classification just as a reaction between these strands. These things are fairly slow. So this process, if it's done at nanomole concentrations, you're looking at a few hours to fully saturate to the equilibrium state. So we first wanted to revisit our results from the DNA and check whether what the RNA model tells us. So far, what we did for RNA is also valid for DNA. So one of the things that the model allows us to do is to sample the free energy landscape. So the free energy is a function of the number of base pairs that you create between the invader and the substrate. So you see that as you form the base pairs for the toehold, you just decrease in free energy because you are getting new hydrogen bonds, so it's a favorable operation. Then you get to the state where you are fully bound to the toehold. Now you start the displacement reaction. So the green one starts eating the base pairs from the blue, and the blue starts to get kicked out for this particular image. Sorry about switching the color code from the previous slide. So the number of hydrogen bonds stays the same, it's fixed, but you nevertheless see about 2 kT penalty. So this system is actually less favorable to be in the strand displacement regime than to be just bound to the toehold. This is an entropic effect because you have these 2 strands hitting each other, so the conformational space they can access is reduced by the fact that the blue and red are clashing with each other. You can better see it here that basically the blue one, when it's just bound by the toehold, it has this entire conformational space that it can occupy, whereas here the blue and green start to compete for the same space of configurations. And this is actually something we predicted, and we could actually, our colleague could do the melting experiments and indeed see the change of melting temperature due to the conformational predicted penalty there. So that's why we have this increase in the free energy. And of course, once you kick out the other strand, then the free energy go down again. Another thing that the system has is that as you increase the length of the toehold, you increase the speed by orders of magnitude until you saturate at maximum speed. So it's of course sequence dependent. So in this plot, we just use the average of AT and CG strength. So we use the average strength model, so we don't have any sequence dependent effects. And this was one of the big questions that our original collaborator had for us for the DNA, but it's the same for RNA. Why is there the speed up and then the saturation? So what happens is that if the toehold is too short, you bind, you try to displace, but you also can fall off. Then you have to diffuse for a long time before you get a chance to bind again and start competing. But once you make the toehold stable enough, then every time you attach by the toehold, you will very unlikely fall off. So in that case, the only limiting step is the time it takes you to diffuse and find a binding partner. And then at normal concentration, at least, the time it takes you to kick out the incumbent strand is negligible compared to the time it takes you on average to diffuse and find the binding partner. So this is why the speed doesn't increase anymore. This is a kinetic simulation from forward flux sampling. The new thing we found for RNA, which wasn't present for DNA, was that it does matter whether you put your overhang at the 3' end or at the 5' end. The difference is that RNA forms this A-form helical shape. So if you place the toehold at the 5' end, the DNA invader will form very strong cross-linking interaction with the incumbent strand, which isn't present if you place the 3' end toehold because the cross-linking interaction is asymmetric. So you only get the stabilization by the 5' end. We predict about 2 to 10 times faster displacement if you place the toehold at the 5' end. It might not sound too much, but there is multiple of the pharmaceutical applications of RNA strand displacement actually rely on some trigger strands, such as, for example, mRNA, which you want to kick out some other strand as a signal. And there, they are really optimizing every single base pair. So having some desired direction to be 10 times faster is actually of importance to them.
So your rate here is really coming directly out of the simulation.
Yes, so this is just relative rate. So I set the rate of the whole length 1 to be my 1.
But you use reactive flux sampling, but it's not
Or flux sampling.
It's not a brute force. Right?
It's not a brute force.
I think that's the question.
No, I was just wondering because the difference is 1,000,000 times, right? More than that. So you should be able to sample all this. You should be running some simulation 1,000,000 times longer and to get.
Yeah, so it's for flux sampling, and for each interface we have.
But I don't think people know what forward flux sampling is.
I see.
Like,
Just look at the rate of probability of going forward.
Yeah, so one way to do it, you would just run millions of brute force simulations and average the rate. This will take a long time, so that's not what we are doing. So we split the reaction into different steps, and for each step we run like several thousands of simulation. So we measure the rate it takes you to bind to the toehold, and then we measure what is the probability if you are bound to the toehold that you will make 3 base pairs.
yeah,
And
something like milestone.
Yeah, milestone.
Yeah.
Thank you.
so, okay, so we discussed this actually during the dinner. It's not easy to be a theorist and coming to experimentalists and asking them to do things for you. The nice collaboration is experimentalist comes to you and asks you, oh, my system, I don't understand it. Can you get some insight? So I never was able to get people to verify some of the strand displacement predictions. So when I started my own lab, I had a student who was also sort of interested in doing experiments. So we first finished all these simulations. And I said, well, why don't we try just to mix few strands in the lab? Because I saw people in the nanotech had very easy time setting up their experimental labs. The trick they didn't tell you is that RNA is really hell to work with because it gets degraded all the time. It took us just 1 week to figure out that somebody didn't wipe clean their desk, which is why all of the RNA got degraded even before we started the measurements. So the results from the experiments, which was eventually published, are sort of incomplete. The reason is that at some point my student refused to enter the experimental lab anymore, and he said, just publish what we have now, and that's it. So I couldn't force him to do anymore. But basically, what we are interested in was confirming following predictions. One is the position of the mismatch. How does it affect the speed of the reaction? Confirming that the length of the hold indeed causes the exponential speed up of the reaction. And then we were interested in the hybrids, like what happens if DNA invades RNA duplex, or what happens if RNA invades DNA duplex, or etc. So I didn't show the simulations for the effects of mismatches because we didn't do them for RNA, but others have done that for DNA. And they actually showed that the system is the most sensitive if the invader has a mismatch with respect to the substrate close to the toehold region. Because it's a kinetic process, and you bind, unbind, you bind, unbind. If the kinetic barrier you have to overcome is very close to the start of the displacement, it really kills you the most. And it indeed is what we saw happening here. Like the rate, this is actually a measured rate per molar per second. The rate is the slowest for the mismatch closest to the start of the branch migration. And the further away you are from the branch migration, the less important the effect is. This was confirmed by simulations before. I didn't show the data. And then we were interested what happens with the story which should be specific to RNA only, the difference between 5' and 3' end. So this is the thing which didn't really measure the data really well. So for one nucleotide, we waited several hours and we still couldn't fit the curve. So we couldn't equilibrate the experiment for just one nucleotide toehold. So the data are actually not interpretable. For having toehold like 3 nucleotides, the experiments predicted the effect to be other way around. So for a reason we don't understand, 3' nucleotide toehold was faster than 5'. And only for the 6 nucleotide toehold, we did see the effect that the 5' end should be, in our case, it was about 20 times faster, even though the simulation would say it would be 10 times faster. It was rather unconclusive. Reviewer wanted us to redo the experiments with different sequences, which would be the correct thing to do, but that was the point where my student had a breakdown. So we respectfully apologize to reviewer, we just can't do everything. And it worked. Well, we just published this and said, it is not agreeing with what the simulations really predicted. Simulations could be wrong. There could be some issue with the particle sequence we chose. We really made the effort not to form any secondary structure. But the correct way to do it would be to try 6 different designs for the fixed toehold length and compare them. But these strands are also quite expensive. So it was a few thousands just for a few RNA strands. So just to be honest with you that, we are not entirely sure what the story is for the toehold location. But we did confirm that there is the exponential speed of it increasing the toehold length. The mismatches work. And one of the things which was actually of interest to biologists is what is the effect of the hybrids? What happens if RNA invades RNA versus DNA invades RNA? So this is not simulation, this is just empirically reproduced free energy profiles, what we would expect to happen. So we know that, yes.
Are the oxRNA and oxDNA?
No, so we do not have DNA RNA hybrids. So we sort of have like intuition from the DNA and RNA simulations what the free energy landscape should look like. So then we just do these published free energy models that people measured in experiment, what is the stability of different base pairs. And we sort of sketched what the free energy profile we would expect. So if you have RNA displacing RNA or DNA displacing DNA, you don't change the free energies. It should be on average pretty much straight line. But if you have DNA replacing RNA hybrid, RNA-DNA hybrid is less stable than the corresponding RNA sequence. So you would expect an uphill climb, unfavorable for the DNA to invade and replace RNA. And if RNA is invading DNA duplex, then you would expect to be slightly downhill because RNA-DNA hybrid on average
your hand
slightly more stable than the corresponding DNA-DNA hybrid. And what we saw was, at least for the experiments we had, sort of compatible with what you would expect from this intuition. For the 3 nucleotide toehold, RNA invading RNA was the fastest. Then DNA evading DNA was second, and RNA evading DNA was the slowest. Now, I did say that the RNA evading DNA should be a slightly downhill climb, but there's another effect at play that you have a B form helix, but the DNA RNA hybrid should be A form helix. So we expect that you are actually rearranging from B form chain to A form chain, which might slow down the reaction. And for the 6-nucleotide toll, we again saw that RNA evading DNA was indeed faster than DNA evading RNA. Again, we suspect that if you have these hybrid systems, the fact that you have to rearrange from B form to A form helix does slow down the process a little bit. So this was mostly experimental work. Why I was really interested in these DNA RNA hybrids is twofold. So one thing, there is increasing evidence, but I wouldn't say consensus yet, in the field of RNA biology, that the strand displacement reaction takes place in many natural systems. So my friend at Northwestern University, Julius Lux, has done structural studies of RNA as it's being folded. What his lab does, it can put a roadblock and stop synthesis of RNA at a certain point, and then he does shape experiments to identify the secondary structure. Now he sees if you stop the transcription at 109 bases versus 110 of the same RNA, it has very different shapes. And what we suspect is happening, we know that the strand displacement reaction is very sensitive to the length of the tow hole. So having one extra base pairs would speed up the strand displacement by a factor of 10 at least. And what is a plausible scenario is that basically this region acts as a tow hole where the loop binds to it, the rest of the loop displaces this base pair, and then this exposed loop replaces this second base pair step. So it'll be like internal displacement within the molecule. I have simulated one trajectory along this pathway, but it was a biased simulation. So I really forced the simulation to follow the path I just described to you. If I would just put the molecule in the simulation box and wait it wouldn't spontaneously happen within my life. Well, maybe yes, but not within the lifetime of a graduate student's PhD thesis. So I wouldn't say this is a proof, but this is at least showing it's a plausible pathway by which these rearrangements can happen. And the second case where I think this is also happening is CRISPR-Cas9, where we have the guide strand, which is the RNA, which targets the region on DNA genome which you want to cut. Of course, off targets are really things you want to avoid. You don't want to cut the DNA in the wrong place. And people have done many mutational studies where they try to introduce mutations in the guide RNA strand and see whether it still cuts or not. And they did see that the probability that the guide RNA falls out and the cutting doesn't happen is very high if the mismatch is very close to the binding site where the protein Cas9 binds and then the RNA starts invading. So this means that it basically falls off, but whether the mismatch is very towards the end, it doesn't really matter too much, and the CRISPR-Cas9 cuts anyways, even if the guide RNA strand is still perfectly complementary to the DNA strand there. And people know that ATP is not required for this process, so it's all compatible with the effect that this is sort of strand displacement reaction where RNA invades a DNA duplex. Again, not necessarily accepted wisdom. My colleague, when he wrote a review of strand displacement, a reviewer made him take out the sentence when he said, I think this happens for CRISPR-Cas9. But now Jennifer Doudna started citing our work on strand displacement in some of her papers. So maybe some people are warming up to the idea that we can explain the CRISPR-Cas9 action partially through strand displacement. Of course, there's also the protein around, and the displaced strand starts interacting with the protein. So the energy landscape is more complex than what I showed you, but it might partially explain what we are seeing there. So this was the RNA strand displacement reaction. Second example of applications of our modeling, which I want to show you, is constructions of metamaterial lattices by self-assembly. So this is one of the directions I mentioned for the field of the nanotech is taking, is to be basically used as a scaffolding to produce interesting shapes in 3D at nanoscale. This particle lattice, so this is a unit cell of the lattice, so you would imagine that this box fills in 3D space. And this particle arrangements of the beads is called a pyrochlore or tetrastack lattice. It is in the same family of lattices as the cubic diamond lattice, which is slightly more famous. It's been calculated in 1990s. If you were able to make the cavity size comparable to the wavelength of visible light, and this made out of silicon, then it would basically act as a metamaterial for visible wavelengths, which means it is transparent for certain wavelengths and it's reflected for others. Mathematically, it is equivalent to the behavior of semiconductors for electrons. So the idea is if you can do this for photons, you can use it as a transistor for light. There are very interesting photodetector applications or optical computing applications. And indeed, if I take the abstract of Alexei Tachenko's paper from 2018, it says that self-assembly of cubic diamond lattices is a long-standing problem of self-assembly. It's often viewed as the holy grail of the self-assembly field due to the challenges that it presents, as well as thanks to its potential as a step towards manufacturing of photonic bandgap materials. So people have been trying for a long time to get these type of lattices assembled. Why is it so difficult, and how do we do with DNA? So a lot of the community effort has been based on using colloids coated with single-stranded DNA, so we would have like sort of uniformly sticking beads to each other. Our approach is to use slightly more complex shapes. So we would abstract the assembly units as spheres with sticky ends, which you would call patchy particles. And the idea is to find interaction between these patchy particles so that they could be realized with DNA nanostructure. This is a wireframe icosahedron. It has these single-stranded overhangs. And if the two single-stranded overhangs are compatible, we would say that these patches can bind to each other. And we use oxDNA simulations to make sure that you can actually arrange these icosahedrons into the lattice geometry and that it would be mechanically stable. Now I spent half an hour talking about oxDNA and oxRNA models. The dirty secret is that as coarse-grained as they are still too slow to just throw these icosahedrons into the box and see anything happen at all. It's still too slow for that. So we need to coarse-grain one level up, and we have to really just work with the beads with sticky ends, patchy particles, because then we can throw them in the box, let them diffuse around and see whether they bind to each other or not. So we did exactly that. We start at the unit lattice with 16 beads, and we designed the interactions to be compatible with each other. So the simplest thing you can think of is that, you know, the green binds to green, which would correspond to self-complementary DNA strands. But if you want to exclude self-complementarity, you can also come up with different interaction rules. So we had a few designs. For the simplicity, let's just assume that each sticky end binds to each sticky end. What happens if you throw them in the simulation box? They diffuse, and after a few hours, so if you would initialize the system in the perfect lattice, it would hold it, because that is a plausible free energy minimum of the system. But if you actually run the simulations and see what is kinetically happening, what you discover is that there exists an alternative free energy in the free energy minima in the system, and that these bastards tend to form like these long filaments, which I would call kinetically preferred alternative free energy minima. And no matter what we did, any solutions we could come up with would always be able to form these things. So what we really needed was an efficient algorithm to search through the design space of all the possible interactions that can exist that would satisfy the target lattice, but also avoid the ability to satisfy the competing structures. Now, I spent one very long, frustrating summer going through many, many different algorithms. I don't have time to explain what we ended up with, but the basic idea under the hood is that you transform your design problem into a set of binary variables. So you basically encode all the possible pairs that could exist in your system. So you say to the algorithm, I want to design this lattice. This like says that particle in position 1 interacts with particle in position 0 through the sticky end number 2 and 5. And I want the algorithm to tell me which patch should be compatible with other patch. And there is a combinatorial explosion of all the possible interactions that would exist. So I cannot test all of them in simulation. The simulation still takes a few hours to run, which I don't have time for. So there is a way that you can encode this as a binary problem called SAT. It's a well-studied problem in computer science, and people have designed algorithms which can search the design space very efficiently. I can talk about it offline if you are interested in more details. The good thing is that we can throw possible design matrices. It tells us whether it satisfies the target shape, and we can quickly test whether it can also satisfy the undesired shape. If it also satisfies the undesired free energy minima, we throw out the solution out of the window and we try a new one. And this way we are able to actually prove rigorously. You can never assemble the lattice I was showing you with just one particle type. The minimum you need is 2, and this is the interaction matrix. So if you throw them in the simulation box, within a few hours, they self-assemble into the desired lattice. So then, of course, the next step would be to do this with DNA. So now we know which strand should interact with what other strands, so we can assign them complementary sequences. And then we use the oxDNA model to optimize the length of the overhangs and their positioning to make sure that they hold the desired target shape lattice. So we would just use oxDNA as a sampling simulations to equilibrate the structure, which contains around 2 million nucleotides, and make sure that we indeed hold the whole geometry. So again, I faced the issue that no experimentalists wanted to do an experiment that we suggested. Fortunately, or maybe unfortunately for the student, I showed these simulations to one program officer who somehow liked them and decided to give us a grant to a theoretician who never held a pipette before. So I was able to hire an experimental postdoc and get money for the experiments. So 3 years later, this was actually only 3 weeks ago, we did manage to image with scanning electron microscope what looks like the lattice we want. But this is still very fresh data, so still research in progress, but we have been getting very frustrated for a long time. We went through many different designs. The icosahedron I showed you never worked. We are still not entirely sure why, but the second design, which we made more rigid, did produce the desired pattern. And last week, we also do some super resolution imaging with DNA paint. So we are sort of trying to image at least the top 2 layers by fluorescently labeling the DNA origamis and trying to reconstruct the 3D shape. Not going super well yet, but it's another method we are trying to develop to characterize the surface. And we also have another design, which is slightly more rigidified icosahedron, which now uses 4 helices per edge. Again, this is the simulation. You see in the scanning electron microscope data, you see sort of some straight lines, but the pattern is not yet as clear as it was for the previous design. So we are still debugging the process. Okay, we also did the design for the other lattice. I will skip it in the interest of time. So the other highly coveted system, the Cube diamond, we also have a tentative solution identified with the same method, which work in simulation, and we have experimental design, but we haven't moved on to realization of this particular design yet. And what I wanted to talk about is like the system of the tools that we developed. So basically all the tools that we have put out there in the past 4 years have been developed for this tetrastack project. Because we didn't have any data for 4 years, we just kept publishing the tools to show some activity on the project. So one of the big challenges we had was that we really needed some interactive design tool where you can load a nanostructure, make modification, and check whether the simulation indeed holds the shape you desired. So this is a tutorial video of the typical workflow we have developed when we want to characterize nanostructure. It is also for other projects, but basically, if we experiment in the nanotechnology, use a tool which is called CAD-Nano, which is fairly difficult software to use, which is very high steep learning rate. You basically draw the helices in a 2D plane. So for us, we want to run simulation is rather unfortunate because we don't actually have the correct 3D positions of the nucleotides. So we sort of guess what are the 3D coordinates based on the helical connections. You can import the structure into this online tool, which we call oxview.org. Inside the tool, we implement very simple rigid body dynamics algorithm so that it just treats all the compact regions of the nanostructure as rigid objects, and the overstretched backbone connections, which if you try to start simulations, it would immediately explode, are treated as springs which push together the shape. So the whole goal of this exercise is to transform the structure from their design software into a format which could be simulated and interactively edited. So once you have the structure reasonably looking, we have a dedicated GPU card on our server to which you can connect, and you can run the simulation that it sends what you have in your browser to our server. It runs simulation for 1,000 steps and sends back to your browser the updated configuration. So it is not meant for production run, but it is meant to get some direct feedback on some design choices you made or for relaxing the structure. So for example, you might want to see what happens if you cut 2 bones in the structure somewhere. And that's something which within a few minutes on a GPU simulation, you would already see the effect on the shape of the structure. So in this case, we know that the structure is still not perfectly happy. You see like these 2 nucleotides, which would be nearest neighbor on the strands, are too far apart. So you see very overstretched backbones interaction. And the simulation is run in a special regime where it relaxes the force gradually. So sort of like the initial equilibration that you do when you simulate crystal structures, for example, it's implemented for the purpose of these structures. So this way you can not only relax the structure, but you can also prepare them for production runs. Besides what I showed you was sort of only DNA and RNA, we have a extended version of the model which also allows representation of proteins as an excluded volume rigid body. So we have explicitly developed this for DNA hybrid nanostructures. So there are people who like can attach covalently DNA strands to proteins. So in this case, we are able to incorporate the protein, load the RNA DNA nanostructure around it, and prepare it for simulation. Now, some of the ongoing projects are, for example, loading spike protein from SARS-CoV-2 virus and designing a nanostructure which exactly positions the binding aptamers onto the spike protein. So it's very rudimentary, but we are not trying to predict new interactions between DNA and protein. We need to have some inputs from the experimentalist colleague who says, you know, I know that this aptamer binds to this particular amino acid, and then we can try to design the nanostructure in such a way it will really form it. This is another project where we tried to make a protein DNA hybrid and fit the cryo-EM profile with it. In this particular exercise, the goal was that the simulations predicted that if you have between 0, 1, 2, 3, or 4 proteins in the structure, it deforms it slightly differently. But wanted to see whether by the deformation of the cryo-EM image, you will be able to predict how many proteins were loaded in it. But unfortunately, the nanostructure the cryo-EM was so noisy that all of the 4 models fitted. So the conclusion there was we don't have enough resolution on the cryo-EM to distinguish between the 4 stages, even though the simulation predicted each of the cages should be deformed slightly differently. Another feature that the interactive tool has is that it is programmable. So you can basically write a program which automatically edits the structure for you. So one of the applications we did was like, say, automatically attach on every available overhang extra 20 poly-T base pairs or single-stranded poly-T. As I said, the whole infrastructure was preferably developed for these crystal structures. So what we mainly use it for was that we say, this is my unit, and copy and paste the unit into the target lattice, glue them together, and export them for simulation. So this was all like 2 programs in JavaScript. In terms of the resources that we made for the community, we encourage everybody to upload their experimental or simulation structures to nanobase.org so that other people can download them, simulate them, and edit them as they please. And for the most common use cases, which is using the oxDNA or oxRNA models just for sampling simulations and getting the idea what the mean structure and the flexibility of the structure looks like, we have the free public web server where people can upload the structure, run it for 4 days, and get a flexibility analysis or analysis of distances between different regions of the structure. evaluate it from there. So how am I doing with time? I'm 5 minutes. Okay. So I have like 2 other examples of our past applications of the model. This is from the more friendly use when experimentalist approaches doesn't ask us if we can get some insight into the system. So this is work by Michael Famolok's lab in Bonn. He has what he calls an ATP-powered leaf spring engine. This is a DNA hinge. He has a T7 polymerase, which is covalently cross-linked to this bottom DNA brick. And there's a DNA which codes for a particle RNA. And there is a stop codon later on. So the polymerase goes along the DNA until it hits the stop codon, then it falls off and binds again to the start and starts transcribing. So it basically goes in the cycle opening and closing. And we were approached by our colleague whether we could first simulate the system and could probe the effects of few design choices he was interested in. So we used the setup tools I showed you to set up the system. We were also able to help him with the design where he has one hinge opening and closing, which drives opening and closing of the hinge, which is connected through it through overhangs. So we like helped design the position of the overhang so that these two can form a dimer and form the driver and follower system, where just this one is ATP powered and the other one is open and closed through the hinges. And some of the questions you are trying to address is what happens if you introduce a NIC into the duplex, because he did see that introducing the NIC had an effect onto the opening and closing rate of the system. And we also wanted to probe different designs of the hinge region, in particular the effect of secondary structure in the single-stranded part of the design. So the main conclusions was that in our simulations, we of course don't have the T7 polymerase, so we just apply a force of 17 piconewtons and pull the 2 ends together. We didn't see any effect introduced if we completely deleted the bridge or introduced the NIC. It didn't have any effect on the equilibrium angle of the system, and it didn't have any effect on the kinetic of opening and closing of the system either. Whereas they did see an effect in the experiment, so we assume that the effects are due to the supercoiling, which affect the polymerase, but there's nothing structural that would be affected by the presence or absence of the bridge or NIC inside of that bridge. The only thing which indeed had a big effect in the simulations on both the equilibrium angle or on the rate of the opening and closing was the presence of secondary structure hairpins in the single-stranded region. So in the simulations, we can explicitly design the sequence not to form any hairpins, which they couldn't do easily for the experimental design. And we see that the rate is different whether the hairpins are present or absent in this hinge. So the conclusion we could give them was that all the effects you see due to the NICs in the bridge are just due to the polymerase effects. And if you want the rate to be faster, you should just delete, re-optimize the secondary structure so that it's as single-stranded as possible and doesn't form any secondary structure in the loop. And very last thing, this is just a theoretical work. It was inspired by diagnostic work, which is done by Paul Rotemund in Caltech. So this is the original DNA origami tile. This is just a 2D DNA layer. And for one of the diagnostic applications, they basically wanted to decorate the origami with single-stranded overhangs, which would carry proteins that would be binding to the target. Now, the system wasn't working very well, and the behavior they saw was compatible with the fact that it wasn't adopting a flat structure. And we introduced the overhangs in the simulations. We actually saw the structure crumble up. And the reason is entropic, because again, if you recall my argument from the strand displacement, the overhangs can clash with each other, but also the overhangs tend to clash with the surface of the tile. If you bend the tile, they have a more conformational freedom accessible to them. And that's basically what causes the bending. You can use the simulations to obtain the entire free energy profile. So we look at the free energy profile as a function of the distance between the edges. So if it's 60 nanometers, that means basically completely open flat structure. But as you keep increasing the density and the length of the overhangs, the most favorable state shifts towards shorter distance, which corresponds to basically seeing the bending. And of course, in the simulations, we also have the option to switch off certain interactions. So if we switch off the interactions of the overhang with the tile itself, we see that basically it returns from preferring 25 nanometer distance to going back to 6 nanometer distance when we switch off these interactions. So it confirms the hypothesis it is the excludable interactions with the surface of the tile that drive the bending of this system. Okay, so that concludes a random walk through some of the recent applications of our software tools. The work I presented, the metamaterial lattice, was done with my colleagues in Rome, Lorenzo, John, and Francesco, and in Venice, Flavio. The original model and some of the subsequent applications, they were developed in Arlui and John Doy's group, together with Tom Aldridge, who's now professor at Imperial College. The leaf spring engine is done by Michael Femlock in Bonn. I didn't really talk about our collaboration with VG on some optimistic modeling of holiday junctions. And my collaborators at ASU, who were kind enough to let me use their lab as I please, as long as it's my student holding the tubes. And those are actually the people who did the work. So Joachim, Eric, and Michael have developed all the online interactive tools for the designs. Hao is the brave student who did all the experiments I presented to you. And Jonah and Matthew are working on these 2D tasks that I showed at the end. Thank you very much.
Thanks. Claudia had a few questions. I'll start. Okay. So you said that you get what 2 million nucleotides on a single GPU card.
Yes.
That's how many particles? It's like
One particle is one nucleotide in our case.
okay.
So it's 2 million particles.
Well, that's still impressive. What's the limit to scaling up?
So the limit is the actual GPU memory.
Memory. So
And so if you use 2 million nucleotides, it's almost 12 gigabytes of memory, which is used by the cell list. Now you can, of course, by changing the size of the cells, you can reduce the memory usage, but then you slow down the actual simulation. But it's the memory which is the main bottleneck for scaling it up.
otherwise it scales linearly.
Yes, so it scales linearly with the number of particles.
Any other questions, please?
So I'm working on DNA nanotechnology still, but I know little about the computational chemistry. So all of my question is about how should I properly use the software to direct my experiment. So my first question is when you talk about the salt concentration, are we using the monovalent cations or not?
Yes, so basically the parameterization of the biochemical interaction was done on data for different monovalent sodium concentration.
Okay.
So now when people talk about magnesium, so if you are just interested in the thermodynamic effects, Felix Retort in Spain, he did experiments where he took a DNA hairpin and he unzipped it and measured the free energy difference between open and closed hairpin by displaying experiments. And he did it at different magnesium, at different sodium concentrations. So he found that if you have X concentration of sodium, it behaves just in terms of the the thermodynamic stability as X divided by 100 magnesium. So I would say if you have this concentration of magnesium multiplied by 100 and use this as the parameter for our sodium concentration. Now there are other things that divalent salts can cause, and those things we would not capture. But if you are just interested about the thermodynamic stability effects, I would just use this Felix Retort's conversion formula for the thermodynamic stability. We do not have any explicit ions though. It's all implicit solvent basically. So in this respect, it's very simplistic. If you have some RNA structure which is stabilized by magnesium somewhere, no way we could capture that.
And the second question is when you are talking about the 5' and 3' and you test the different like the ratio or the rate or the difference between the 5' end and 3' end. But when the oxDNA actually doing a calculation, I feel like they only like calculate the pipette stacking, the hydrogen bond, all the things, the distance. But how the software differentiate the 5' end and 3' end?
So basically, each base knows who's the 5' neighbor and 3' neighbor. And for
Yes, yes. I know, like it's the atomic level differences, right? And
so
the
I mean, when I have the DNA strand or the RNA strand, I mean, we have a visualization option which like draws an arrow in the 5' direction or 3' direction. But basically, each nucleotide knows who is his 3' neighbor, and it knows who is his 5' neighbor.
and
But the effects of the difference between 3' and 3' end, they are an emergent property.
Okay.
Because you know all the potentials in the model are pairwise, and they are designed in such a way that when you form the duplex, the RNA duplex has this inclination here.
Okay.
Once you have the inclination and you have the invader coming to the 5' end, the cross-stacking interaction has an ideal equilibrium distance at which it likes to act. If you're on the A-form helix, these two guys are closer than these two guys. So the cross-stacking interaction will only act between this pair, and it's going to be extremely weak between this pair. So that means if you bind from here, the invader strand gets a cross-stacking interaction with this guy, which stabilizes it by some number. Whereas if the invader comes from the 3' end, the cross-stacking interaction which is strong is already present in the system because it's between the overhang and the incumbent. But the new cross-stacking interaction which you form is the asymmetric distant one. And that is going to be very weak compared to the one which you can form here.
Okay. And the last question is, when you talk about the protein and DNA interaction in the last part,
Yes.
do you somehow like define the interaction between the DNA and protein, or the software could?
Yeah, so it has to be explicitly defined. So the only interaction we have between the protein and the DNA is excluded volume, which basically says they cannot overlap with each other.
Okay.
But the fact that the aptamer binds to this particular amino acid has to be explicitly made. You basically create an interaction in the design tool and bind these two together explicitly.
Okay.
So it can be used to predict new interactions, but you need some experimentalist input that tells you, you know, this is where it has to bind, and we explicitly put it in the simulation.
Okay, so basically, I tell the software that the protein can interact with the DNA in this way, and then they can try to like do fix the other part. Does that mean? For example, like the protein has 3 binding sites, the
Yes.
DNA just like the model you just showed. And should I just tell the software that I have this kind of binding, and they can do the other 2 sides?
No,
or-
we have to do it for each side explicitly.
Okay.
Currently, in the future, we might make it more flexible. But the way the currently stands, you have to initialize on the interactions before you start the simulations.
Oh, I see. And
Or-
they won't separate.
They won't break
Oh,
up. In the future versions, you will enable the option to break up, but currently it's all fixed.
Yeah,
It's
I
mainly made for when you want the protein to be part of the nanostructure, so that you can design the nanostructure to exactly match the binding sites. Was the application we had in mind here.
Yeah, that's very useful. Thank you.
Any other questions?
So I just want to make sure that I miss it. So you showed this beautiful movie where the strands come together.
Yes.
But I can imagine for those very complex structures that you have, you cannot do this automatically, right? There are tons of metastable.
No, so
How do you tell the system to come together the same way that you want it, right?
for these DNA origamis, for example,
No, but this one is a brute force.
yes, for these DNA origamis, we initialize the simulation already in this shape.
Oh,
We don't just throw the strand
Oh.
wait for them.
so you design it like this?
Yeah, so people have these design software I was showing in the tutorial video. So they have rudimentary, not that easy to use software, they sort of like draw these things in 2D plane. And we convert it into a 3D shape from that
But
data.
it's a predictive.
aspect of the simulation.
So in many cases, the most typical thing that experimenters want to know is what is the shape that the structure actually adopts. They have something in their mind when they draw it, but maybe the distance is too short and this thing crumbles up. Or maybe they want to position 2 nanoparticles here and here to have some plasmonic effects, but they tend up to be further apart than they anticipated. So these are the things we just don't have capacity to work with people anymore, which is why I created the server and just tell them to upload it there and run it themselves.
So that brings me to my next question. So you can download and run the simulation.
Yes.
Yes.
Which requires somebody to know how to use command line, which is sometimes already a high bar for people who don't have any physics or chemistry students. So for them, you have like this graphical software where they can do it. It is true, I would want to have a software where I can assemble these things from just the single strands. Unfortunately, this software, this representation level is still too slow. I had a colleague who did PhD after me. His PhD was to actually study the kinetics of assembly of these structures, because some designs have like yield only 2% and some have designs 95%. So there are important kinetic effects which go into their self-assembly, which our model cannot capture by brute force. So he basically had to run forward flux simulations, which basically means that for each strand that was added, he would have to run certain pathway thousands of times and get the statistic of what is the probability if you are bound here, that you're also bound here. And he took him 4 years, and at the time, nobody in Oxford had GPU code they could use. So we had 80 GPUs just for him for 4 years to get all statistics. So it's still, unfortunately, not at the level he would like it to be in terms of the systems we could study.
All right, well, many thanks. And that one was